So, usually in oncology, right, a diagnosis requires you to actually get a physical piece of the enemy.
Right, exactly. A scalpel.
Yeah, a biopsy. You need to extract the tissue to understand what you're up against. But what if the exact 3D microscopic architecture of a tumor, I mean, its walls, its guards, the vulnerable core, what if all of that could be mapped without ever actually touching the tumor?
It sounds like sci-fi, honestly.
Right. Like, what if you could reconstruct the entire geographical battleground just by analyzing the debris floating in a routine blood draw?
Which is wild to even think about.
It really is. And that is the premise of a massive leap in tumor biology published in May 2026 in Nature, which details a framework called spatial ecotypes. So, welcome to the deep dive. We're so glad you're here with us to unpack this.
Yeah, and this paper, I mean, it represents a fundamental shift in how we actually perceive cancer.
Because we've been looking at it kind of wrong, right?
Well, for decades, yeah. We've treated tumors like these chaotic bags of mutated cells. Just a mess.
Yeah.
But this research proves they are actually highly structured, geographically rigid ecosystems. And more importantly, that we can read that geography remotely.
Okay, let's unpack this. Our mission for this deep dive is to decode those cellular ecosystems, scrutinize the machine learning pipeline that actually found them among millions of data points, and look closely at how this new tool, Liquid EcoTyper, is predicting immunotherapy success.
Right, without ever needing a scalpel.
Exactly. So if we accept that the tumor isn't just a biological blob, but like a highly organized walled city, the immediate problem is that our traditional methods of studying that city have been practically blind to its layout.
Precisely. Historically, we face two massive hurdles. First is sampling bias.
Right, the needle problem.
Yeah, a standard core needle biopsy only captures a tiny localized fraction of the tumor. You might hit a necrotic zone and think, oh, the tumor is dying.
But you're completely missing the aggressive invasion happening like 2 millimeters away.
Yeah. Exactly. Imagine you're a general looking at a battlefield. If you only see 1 square foot, how can you predict the outcome of the war? You can't.
You really can't. And the second hurdle is what happens when we use bulk RNA sequencing. I like to call this the smoothie problem.
The smoothie problem is a great way to put it.
Yeah, because you take the biopsy, grind the tissue down, and sequence the RNA to see what genes are active. You get a comprehensive list of all the cell types and mutations, but you completely obliterate the spatial coordinates.
Right. You have myofibroblasts and T cells in the blender.
But you have zero idea if they were interacting or if they were on opposite sides of the tumor.
Exactly. So to solve that, the researchers behind this paper turned to spatial transcriptomics, or ST.
Which preserves the location, right?
Yes. It sequences RNA while preserving the 2D and 3D tissue coordinates. But they didn't just run a small pilot study. I mean, they built a staggering data set.
The scale is honestly hard to wrap your head around.
It is. They mapped over 10 million single-cell and spot-level spatial transcriptomes. This was across 132 primary human tumors.
And that wasn't just one type of cancer, right?
No, it covered 10 distinct types of cancer, ranging from various carcinomas to melanomas.
Okay, I want to pause on the methodology there because mapping 10 million cells is one thing, but they integrated data from 6 completely different spatial platforms.
Yeah, that was a massive undertaking.
We are talking 10x, Visium, Merscope, Xenium V1 and V3', Legacy ST, and Visium HD. As someone who follows spatial tech, combining spot-based resolution like standard Visium with true single-cell resolution like Xenium is a logistical nightmare.
Oh, absolutely.
How do they avoid massive platform bias?
Well, it requires extreme computational rigor. If you just pool that data together, the technical differences, like how Merscope captures transcripts versus how Visium captures them, will completely overwhelm the actual biological differences.
So the machine just sees the scanner, not the cancer.
Right. So the researchers had to meticulously normalize the data. They harmonized the spot-level platforms with the single-cell platforms to ensure that when they identified a specific cell neighborhood, it was a biological reality.
Not just an artifact of the specific hardware used to scan the slide.
Exactly.
Which brings us to the core challenge. If you manage to harmonize that data, you are still staring at a spreadsheet containing 10 million data points with X and Y coordinates.
An ocean of noise.
Right. So if you're looking at a microscopic battlefield and you have the coordinates for 10 million individual soldiers, how do you mathematically prove that they are organizing into distinct neighborhoods?
You need a pipeline that can recognize patterns without human bias. So they engineered an algorithm called Spatial EcoTyper.
And this is where the math gets really cool.
It does. It operates in a methodical 4-step framework. First, they define the scale of interaction. They systematically overlaid a grid across these tumor sections, dividing them into spatial neighborhoods, or SNs.
And they used a very specific size for those, right?
Yes, each with a rigid radius of 50 micrometers.
Okay, and 50 micrometers is roughly the distance a cell can effectively communicate using paracrine signaling, right? Like secreting local chemical messengers.
Exactly. So they are essentially drawing a circle around every cell to see who it is actively talking to in its immediate vicinity.
That makes sense.
But here is where they encounter a notorious problem in spatial transcriptomics technical dropout.
Oh, right.
During the physical preparation of the tissue, mRNA transcripts are inevitably lost or degraded. You might have a perfectly healthy T cell in your neighborhood, but the sequencer just misses its key markers.
So it looks like it's not there, even when it is.
Right. To correct this, the algorithm generates spatial gene expression profiles, or SNGEPs.
How did that work?
It averages the transcriptomic data of the nearest cells of the same type within that 50 micrometer circle. It essentially fills in the missing data points by leaning on the immediate neighbors.
So it's rescuing the signal from the noise? Okay, so the algorithm has defined the 50 micrometer neighborhoods, and it has smoothed out the technical dropout. But a neighborhood isn't just one type of cell.
No, not at all.
It's an immune network overlapping with a structural stromal network overlapping with the cancer cells themselves.
Which is where step 3 comes in similarity network fusion, or SNF.
Okay, break that down for us.
SNF constructs patient-specific mathematical graphs of local cellular composition. It takes the spatial gene expression networks for the immune compartment, the malignant compartment, and the stromal compartment.
It layers them.
Yes. It iteratively updates them until they fuse into a single, cohesive mathematical matrix.
It's basically like taking transparent maps of a city's plumbing, its electrical grids and its roads, and laying them over each other to see how they align.
That's a perfect analogy. It quantifies exactly how these different cell types are co-locating in space.
But that fused matrix still needs to be decoded, though. I mean, you have the interaction networks mapped, but you need to extract the repeating patterns that occur not just in one patient, but across all 132 tumors.
Right, which leads to the final step, clustering. They employed Louvain clustering to identify broad community structures within the fused networks.
And then they refine that, right?
Yes, followed by non-negative matrix factorization, or NMF.
Now, NMF is basically a math trick to simplify things, right?
Exactly. NMF is highly effective at taking a massive, dense matrix of data and factoring it down into pure components. In this context, it extracted the distinct, repeating cellular neighborhood motifs that consistently appeared across the entire pan-cancer dataset.
Here's where it gets really interesting. What caught my eye wasn't just the clustering itself, but what happened when they visualized the physical locations of those clusters.
The spatial gradient.
Yes. The algorithm revealed a profound spatial gradient. These neighborhoods didn't just pop up randomly. They mapped a continuous trajectory starting from the deep, hypoxic core of the tumor, radiating outward in specific layers all the way to the adjacent stroma.
Location dictates the biology.
Exactly. Think about your own neighborhood. If an algorithm mapped it, what repeating ecotypes or hubs would it find? A coffee shop next to a bookstore? Well, in the tumor, the physical geography fundamentally drives the behavior of the cells.
And based on that continuous gradient, the algorithm distilled the complexity of those 10 million cells into 9 universal laws of tumor organization.
The 9 spatial ecotypes.
Yes, or SEs, numbered 1 through 9. These are highly specific repeating microenvironments that dictate whether a tumor will grow, metastasize, or die.
Okay, so if these 9 ecotypes are universal across these different carcinomas and melanomas, let's dissect the biology of the most critical ones. Let's start at the tumor margin.
The battle lines.
Right, the margin is where the invading tumor physically meets the host tissue. The paper highlights SE3 as a major hub located right at this border.
And SE3 is incredibly complex. It is defined by a dense accumulation of IgLL5 positive plasma cells, C1QB positive macrophages, CD1E positive dendritic cells, CXCL12 positive cancer-associated fibroblasts, and CD36 positive endothelial cells.
Okay, wait, that is a lot of acronyms. Rather than just listing those markers, we need to look at what they're physically doing in that space.
Fair point. Well, the CD36 positive endothelial cells are laying down the local blood vessels.
The plumbing.
Right. The CXCL12 positive fibroblasts are essentially the scaffolding. And in the middle of this structural network, you have dendritic cells acting as antigen-presenting traffic cops.
So it is a highly active, multi-phenotypic intersection.
Yeah.
The tumor is aggressively negotiating with the host's immune system there.
Exactly. And right next to that negotiation, you often find SE4.
Ah, the bad builder.
Yes. Also located at the margin, SE4 is defined by MYH11 positive myofibroblasts and FBLN5 positive endothelial cells. And this ecotype is clinically devastating.
Right, because SE4 essentially hijacks the body's natural wound healing mechanics. When you get a cut, myofibroblasts rush in to lay down dense connective tissue, while endothelial cells build quick, temporary blood vessels.
But the tumor corrupts this process.
Yeah, it uses SE4 to build an incredibly dense physical barrier, a literal wall of fibrotic tissue around itself.
And that wall has a secondary lethal effect, hypoxia.
Oxygen starvation.
Because the blood vessels built by those FBLN5 positive cells are highly dysfunctional, the area is starved of oxygen. Hypoxia paralyzes cytotoxic T cells, but cancer cells can actually adapt to it.
So SE4 creates this impenetrable, oxygen-deprived moat around the tumor.
Yes. Clinically, high levels of SE4 directly predict rapid progression and severe resistance to immunotherapy. It is deeply immunosuppressive.
Wow. And the margin also houses SE5, which seems to serve a similar suppressive function, but through a totally different mechanism.
Right, the shield. SE5 is packed with FAP-positive cancer-associated fibroblasts and APOE-positive M2-like macrophages.
So while SE4 builds a physical hypoxic wall, SE5 acts as a chemical shield.
Exactly. Those M2-like macrophages actively secrete cytokines that turn off the local immune response.
But the margin isn't purely defensive for the tumor, is it?
No, this is where we see SE7, which is one of the most critical discoveries in the paper.
The border patrol.
Yes, SE7 sits right at the border, but its transcriptomic profile is dominated by high STAT1 expression and genes associated with active antigen processing and presentation.
And STAT1 is a master regulator of interferon signaling. So the cells in the SE7 neighborhood are actively recognizing the tumor antigens, producing interferons, and sounding the alarm.
They are actively trying to breach the wall.
And if they do breach the wall, we look to the deep core of the tumor, where we find SE8.
The special ops.
Exactly. While SE7 is fighting at the border, SE8 is operating deep in the trenches. It is packed with GZMB-positive cytotoxic T cells and CCL8-positive macrophages.
And GZMB stands for granzyme B, which is the literal chemical weapon T cells use to induce apoptosis or cell suicide in cancer cells.
Plus, the metabolic activity in SE8 is completely elevated. These cells are burning high amounts of energy to sustain an active fight.
So you have SE7 sounding the alarm at the border and SE8 actively executing cancer cells in the core.
Right. So if a patient's tumor is rich in SE7 and SE8, their prognosis is significantly better. I mean, if SE7 is guarding the border and SE8 is fighting in the center, which one do you think is more vital for a drug to target? They both are.
And the paper proves this conclusively. They didn't just rely on the primary spatial dataset. What's fascinating here is they validated these 9 ecotypes by mapping them onto 7,076 bulk RNA sequencing profiles from the Cancer Genome Atlas.
And that spans 16 different carcinomas, right?
Yes.
Oh.
By deconvoluting the bulk data using their new spatial signatures, they prove that high SE7 and SE8 universally predict longer survival and a profound positive response to immune checkpoint inhibitor therapy.
Outperforming legacy metrics like the CE9 signature. I do want to push back slightly on the universal aspect, though, because the data showed some fascinating exceptions.
Ah, yes.
In prostate cancer, pancreatic cancer, and certain esophageal cancers, the survival patterns for these ecotypes inverted.
They did.
If SE7 and SE8 are such powerful immune hubs, why did they suddenly fail in those specific organs?
It comes back to the physical architecture of the tissue. Pancreatic and prostate tumors are highly desmoclastic. They naturally produce an overwhelming amount of dense, concrete-like stroma.
Like a natural SE4 wall.
Exactly. Furthermore, these specific cancers often downregulate major histocompatibility complex molecules, so they don't just build a wall, they essentially turn invisible to the immune system.
Wow.
In these highly distinct environments, the standard interactions of the ecotypes are physically blocked and fundamentally altered.
Which highlights exactly why understanding spatial context is so vital. I mean, a T cell is totally useless if it's trapped in a desmoplastic concrete wall. So what does this all mean? If we know that SE7 and SE8 are the active immune hubs we want to support with drugs, and SE4 is the fibrotic wall we need to break down, the ultimate clinical question becomes, how do we monitor this?
Right. Tumors are highly dynamic.
Yeah, they evolve under the selective pressure of therapy. You cannot surgically biopsy a patient's internal organs every single week to see if the SE4 wall is expanding.
And this is the exact clinical bottleneck the researchers targeted with the second half of this paper. They developed liquid EcoTyper.
The blood test.
Yes, a non-invasive blood test designed to map these 3D spatial ecotypes from a simple plasma draw.
Okay, wait, I need to stop you there because this is where the physics of this process seem impossible to me.
How so?
Well, if SE7 is on the outer wall and SE8 is deep in the core, once those cells die, their DNA all gets dumped into the exact same bloodstream, right?
Yes, it does.
It becomes a giant blended liquid soup. So how on earth does an algorithm look at a 1D fluid and reconstruct the 3D neighborhood where the ingredients originally came from?
It requires looking beyond the standard genetic code. When cells die, they shed fragments of cell-free DNA, or cfDNA, into the plasma. Liquid EcoTyper doesn't just sequence the letters of that DNA, it uses whole genome EM-seq to map the epigenetic methylation state of those fragments.
Oh, epigenetics,
Yeah.
the regulatory layer sitting on top of the DNA.
Exactly.
Because methyl groups bind to the DNA strand to physically block transcription machinery, right? Turning specific genes off, while unmethylated regions are left accessible and turned on.
Precisely. And the methylation state is incredibly stable even after the cell dies and the DNA fragments.
Okay, I see where this is going.
Because an SE4 myofibroblast operating in a hypoxic margin regulates its genes entirely differently than an SE7 macrophage fighting an active immune war, their DNA is methylated in completely unique, recognizable barcodes.
So they aren't looking for mutation, they're looking for the epigenetic signature of the neighborhood itself.
Yes. The researchers generated massive amounts of simulated cfDNA data pseudo-bulk mixtures based on the methylation profiles of the 9 spatial ecotypes.
And trained an AI on it.
They trained a machine learning classifier on this data, teaching the AI to read the chaotic soup of cfDNA in the blood and accurately deconvolute it, separating out the unique epigenetic barcodes of the 9 ecotypes.
That is just brilliant. And they actually proved the clinical viability of this, didn't they?
It did, in a pivotal cohort of 78 patients with metastatic melanoma undergoing immunotherapy. They drew their blood before treatment and ran liquid EcoTyper.
And the results?
Startling! Patients who had high levels of the SE7 barcode floating in their blood, indicating a strong Border Patrol presence in their physical tumors, responded beautifully to therapy.
Wow.
They achieved a hazard ratio of 0.33, meaning their risk of disease progression was drastically reduced.
Okay, but what about the patients with the bad wall, the SE4?
Conversely, patients whose plasma was rich in the SE4 barcode, indicating their tumors were shielded by dense, hypoxic, wound-healing walls, exhibited severe resistance to the immunotherapy.
So the drug just couldn't get in.
Right. Their hazard ratio spiked to 2.92. They progressed rapidly, regardless of the treatment.
The critical point here is the comparison to the current standard of care. I mean, right now, liquid biopsies primarily measure circulating tumor DNA, or ctDNA, focusing on mutational burden.
Right.
The logic being that more mutations mean a higher chance the immune system will recognize the cancer. But in this exact cohort, standard ctDNA mutational analysis only modestly predicted resistance.
Because mutational burden ignores the physical reality of the tumor. A tumor can have thousands of mutations, making it highly immunogenic, but if it is completely encased by a hypoxic SE4 stromal wall...
the T cells can never physically reach the cancer cells to kill them.
Exactly.
Yeah.
Liquid EcoTyper drastically outperforms standard mutational tests because it measures the structural reality of the battlefield, not just the identity of the enemy.
How would a simple blood test that reveals the precise 3D architecture of your internal immune response change the way you view a trip to the doctor? It's wild to think about. We have covered immense ground today.
We really have.
We started with the massive logistical challenge of mapping 132 tumors across 6 different spatial platforms to overcome sampling bias. We saw how Spatial EcoTyper averaged transcriptomes within 50 micrometer grids and used similarity network fusion and matrix factorization to distill 10 billion cells into 9 universal spatial laws.
Ecotypes.
Right. We looked at the mechanisms of SE4 building a hypoxic fortress and SE7 and SE8 orchestrating the immune siege. And finally, we explored how whole genome EM-seq can read the epigenetic barcodes in a routine blood draw to reconstruct that entire 3D architecture.
If we connect this to the bigger picture, we are witnessing a complete paradigm shift. We evolved from staring at static 2D tissue slices under a microscope to algorithmic 3D spatial mapping, and now we're leaping into an era of non-invasive liquid biopsies that can read that 3D map remotely and dynamically.
We're no longer guessing what the tumor microenvironment looks like.
No, we are tracking its exact geographic evolution in real time.
It is a remarkable level of precision, and it leaves you, the listener, with a rather profound concept to consider. If an AI can accurately reconstruct the physical 3D neighborhood of a metastatic tumor, its walls, its hypoxic zones, its active battle lines solely by reading the epigenetic methylation fragments floating in a single vial of blood, what other physical structures in our bodies are currently broadcasting their exact blueprints into our bloodstream right now?
That's a great question.
Right. If we could read the 3D map of a tumor, could the same algorithm eventually read the 3D architecture of a developing autoimmune attack or a forming atherosclerotic plaque long before symptoms ever appear?
The possibilities are endless.
The blueprints are in the blood. We just need the right algorithms to decode them. Keep that in mind next time you get a routine lab draw. Thanks for joining us on this deep dive.